So we were here, right? Yeah, I told you that anemia, graveyard of RBC, blood bank, spleen is also called blood bank. Uh, RBC, we did it. Now let's start with the WBC, right? So the second topic is we are going to start with the WBC, and WBC are also called leukocytes. WBC, that is also called leukocyte. Leukocytes. So white blood capsules or leukocyte. Before starting it, I want to share a PPT with you. The WBC are of various size of various type. Here we will study WBC in detail because the number of the question have been asked from the WBC. I will show you PPT. You have to take the screenshot of the same, and you have to jot down it your notebook because that will be very beneficial during your medical examination. So, circulatory system. Circulatory. Look. Here we can see all three type of the cells, the shape of all three type of the cell. So here you can see these are RBC. RBC we have discussed already in detail. And the second one is platelets. Platelets we will do it after the WBC. And you can also see the white blood capsules. So here white blood capsules are different monocyte, lymphocyte, eosinophil, basophil, neutrophil. What are they and what are their characteristics? So we'll discuss after this this PPT. So here you can see the WBC are of different shape. In fact, WBC, if we talk about the WBC do not have any fixed shape. Their shape is variable. So WBC, they have no fixed shape. And one more important thing, their shape is variable. So they have amoeboid shape. Amoeboid shape means they can change their shape. So they can change their shape, they can change their structure. What is the benefit of changing their shape or changing their structure? Yes, there is a benefit of changing their shape and changing their structure. For sake of example, this is a blood vessel. So consider it as a blood vessel. It may be artery, it may be vein, whatever you want to consider. So this is blood vessel. It may be artery or vein. Blood vessel. Inside the blood vessel, there are these cells which we call WBC. So what WBC do? WBC can change their shape. They have a benefit. In these blood vessels, there are certain small creeks. And from these creeks, these WBC can come out. They can change their shape, and by this way, they can leak out from these spaces, right? So because of variable shape, they can come out of the blood capillary. So blood vessels. So what is the benefit of coming out of blood vessels? Actually, here out of the blood capillary, there are some spaces where they can kill the germs, they can kill the pathogen, right? So this is the benefit. That's the changing their shape. And what is their main function? Their main function is to maintain the immune system of our body. They maintain immunity of our body. They fight against the pathogen. That is their main functioning, right? So please start noting now. WBC. Their number varies from 6,000 to 8,000. Their number varies from 6,000 to 8,000 per mm cubic of blood. Per mm cubic of blood. A very easy question that has been asked number of the time in medical examination. They have asked that what is the ratio of WBC and RBC. So what is the ratio of WBC and RBC.
Sir, can you please repeat from their number very strong?
Yes, their number varies from 6,000 to 8,000, 6,000 to 8,000 per mm cubic of blood, per mm cubic of blood. Next point, ratio of WBC and RBC. Ratio of WBC and RBC is 1:600. This question has been asked many times. Keep it in mind. Next point. Rise in WBC is termed leukocytosis. There is one more important term, that abnormal rise in WBC. Rise in WBC is leukocytosis. What we call then when they abnormally rise, right? So write it down. Can you tell me what is the abnormal rise of WBC? Abnormal rise of WBC. What we call that? Note it down. Abnormal rise in WBC leads to leukemia. What is leukemia? Abnormal rise in WBC leads to leukemia. Leukemia is blood cancer. Leukemia is blood cancer. So abnormal increase in or rise in WBC term leukemia. Yeah, leads to leukemia, right? Leukemia is blood cancer. Blood cancer is nothing but what happened in this cancer. Our WBC start eating our own body cell. They lose the sense of that either it's a body part or it's an external germ or any pathogen, right? And they, in spite of eating bacteria, virus, and rest of the pathogen, they start eating our own body cell. That is called blood cancer. Next.
Sir?
Yes.
So in this abnormal rise, what's the number of WBC?
WBC number, they will rise exponential way. They will be 25,000, 40,000, 50,000. That's how they rise. Abnormal disease. Like, you know, suppose that I am like right now I am a fit, right? So right now the WBC count will vary in my body. Approximately, this is the ideal condition, 6,000 to 8,000. That will, it may be 9,000, it may be 5,000 or 6,000 or 8,000. So they will be below the 10,000, right? And 15 or 20 days back, I had a fever. I was sick. So on that time, the number of the WBC, suppose that I got a fever, I got an infection, or I got some disease, right? So on that time, if you check my WBC, the number of the WBC will be 15,000, 16,000, 17,000, right? They may be the number may be up to 15,000, 16,000, 17,000. That is the process of leukocytosis. So leukocytosis happened whenever we get ill or we get sick. On that time, what happened? The number of WBC rise to cure our body, to fight with the germs, right? That's why their number increase. And when we get well, on that time, the number, it again comes down, right? But if in a body, a number of WBC are more than 30,000, 40,000, 50,000, it means the person have leukemia, right?
So more than 30,000, it will be leukemia.
Yes, like 30,000, 40,000. If have 30,000 someone, then it's a leukemia. Even as per my idea, it should be 25,000 is also a condition of, it may be the condition of leukemia as well. Right. Okay. Normally, during the, yes, when a person is not well, it may be 13,000, 14,000, 16,000. That is quite possible. Next, the size of WBC. The
so
size.
Yes. So, for example, like if there is abnormal rise in WBC, and at the same time the person is infected with the virus, then will this equalize, sir? Like,
no, like
the WBC will fight the viruses.
WBC will WBC will rise, but they won't rise like exponential way. They can't go beyond 25,000, 26,000. That is not possible in any disease. because there is some limit, right? Number of division is controlled by genetically in our body, right?
Yes.
Okay. So the size of WBC varies. Size of WBC varies from 10 to 20 micrometer. Size of WBC varies from 10 to 20 micrometer, comma. It also contain nucleus. It also contain nucleus and some cell organelle. Underline it. And some cell organelle. It also contain nucleus and some cell organelle. Next, the formation of WBC is called leukocytosis. Formation of WBC is called leukopoiesis. Formation of WBC is called leukopoiesis. Now I am going to show you a PPT regarding the structure of WBC, and that PPT you have to stick this screenshot. First, I'll explain that PPT. That is very important. I have written all the information which is required for you, and I have put all the information which is required for your medical examination. And you will get the type of WBC. They can ask the type of the WBC, and they have asked, in fact, number of the times. So,
sir, I have a question.
Sorry, yeah, please.
In the previous class, you said that RBC, we cannot call them red blood cells, but we should call them red blood card cells. So what's the reason?
The reason was that RBC do not have nucleus. They do not have any cell organelle. So we cannot call them nucleus because we cannot call them cell, because for a particular structure to call it or to pronunciate is as a cell, there are certain eligibility. The eligibility is that it must have proper plasma membrane, proper protoplasm, and at least it must have nucleus as well, right? But RBC do not have nucleus, right? It do not have any cell organelle. That's why we call it carpel. Let now come to the WBC. WBC do have nucleus, but most of the cell organelle are absent from the WBC. WBC do not have all cell organelle, right? So that's why we do not give them a designation of cell. We also call they are also called as carp cells. Now come to the platelets. Platelets also do not have nucleus and do not have cell organelle are missing. That's why we do not give designation or a status of cell, right?
Okay, sir. Thank you.
Welcome. So now come to the PPT part. Let me show you the PPT. Is it legible to you? Can you read it out? Is it legible?
Yes, sir.
Okay, fine. So let me explain it. So these are the type of WBC. Above here, I have shown you the type of WBC shape: monocyte, lymphocyte, eosinophil, basophil, and neutrophil. Here, we have classified WBC in two categories. So WBC are called leukocytes. So leukocytes have been classified in two categories, right? Granulocyte and agranulocyte, right? Granulocyte and agranulocyte. Agranulocyte do not have granule in protoplasm. So no granule in protoplasm. They do not have any granule-like structure in protoplasm. Granulocytes, they have granule-like structure. They have granule-like structure in protoplasm. So that is the difference between granulocyte and agranulocyte. Further, agranulocyte have been divided in two categories, lymphocyte and monocyte. And the lymphocyte have been again divided in further two categories, B lymphocyte and T lymphocyte. So lymphocyte may be B lymphocyte and T lymphocyte. What are the B lymphocyte? What are the T lymphocyte? B lymphocytes are those cells, those kind of the lymphocyte which are formed in liver. They produce antibody, right? And there are T lymphocyte, they are formed in thymus gland, right? So formation, please keep it in mind that B cells are formed in liver and T cells are formed in lymphocyte, right? T cells are formed in thymus gland. They directly attack. They do not produce antibody. They directly attack on the bacteria or another pathogen. The function of the both of the cell to kill the pathogen. So lymphocyte may be B lymphocyte or T lymphocyte. They are non-motile and non-phagocytic. Means they cannot move, and they are non-phagocytic. Means they do not eat bacteria. They kill the bacteria, they kill the virus, they kill the pathogen, but they do not eat them. Right. Next, they produces antibody and destroy the microbe, toxins, and tumor cells. Tumor cells, underline the tumor cells. This question have been asked in medical examination, that which cells are responsible for killing the tumor cell. So our body have capability of killing tumor cell, right? We do not, rather than like medicines are still not effective against the tumor cells, but our body, even our body own have immune system which can fight against the tumor cells or cancer cells. 20 to 25% of WBC are lymphocytes. Lifespan of few days to year. Star is over there, means the question have been asked. Lifespan is few days to years. Now come to the monocyte. They are the largest of WBC. Important question. They are the largest out of all WBC. The monocytes are the largest one. They are motile and phagocytic. They move here and there, and they are phagocytic, means they can eat the bacteria and rest of the pathogen cells. They engulf bacteria and cell debris. Cell debris means the broken part of the cells, means the excretory part of the cells or any part of the cells. Lifespan is only 10 to 20 hours. Now come to the granulocytes. Granulocytes have been further divided in three categories: eosinophil, basophil, and neutrophil. Eosinophil, they increase in allergy, asthma, and hay fever. It is important. They help in dissolving blood clot. So they are responsible for dissolving blood clot. They are non-phagocytic. They are also non-phagocytic. Similarly, like lysosome, they destroy the protist. So they destroy the protist. Basophils release heparin. Very important. Heparin is anti-clotting agent. We have discussed that heparin is anti-clotting agent. It do not allow blood to clot inside our body. Again, they also release serotonin and histamine. What is histamine? Histamine is a type of substance which is released during the time of allergy, and we get the itching and reactions in our body, in our skin, because of the release of the histamine. Next, basophils. They are least in count, right? Neutrophil, most numerous. That is the very important question. It has been asked. The most numerous. They are 60 to 65% of the WBC. They eat harmful germs. That's why they are phagocytic in nature. Is it clear?
Yes, sir.
Guys, please take the screenshot, and please, I expect that you will write it down in your notebook, and it's very important.
So these granulocytes, they are non-motile?
Which are granulocytes?
Yeah.
Yes. Look, eosinophils, then increase allergy, and have no phagocytic. They are non-phagocytic, histamine. They are also non-motile. Basophil are also non-motile, and they are this one. Neutrophil is motile, and eosino and basophil are non-motile.
So what are B cells and T cells?
B cells actually, B cells are a type of lymphocyte cells. So the lymphocyte cells which are produced in liver, they are called B cells, and the lymphocyte which are produced in thymus gland. Thymus gland is present over here. We'll study in endocrine system, right? Thymus gland is present over here. So those lymphocytes which are formed in thymus gland, they are called T cells, and those lymphocytes which are produced in liver, they are called B cells. Right? Clear?
Yes, sir.
Okay.
So what actually tumor is? Like,
Tumor
why is it not curable?
It's not curable. Actually, what happened when the cancer caused at any part of the cell? So what happened on that cell start dividing uncontrolled manner, right? So cancer is nothing that is uncontrolled division of the cell is called cancer, and uncontrolled division start at particular part. These cells keep on piling at particular part and they make a lump-like structure that is called tumor. After the formation of this collection or lump of the cells at any body, that is called tumor. These cells start competing the other cells. And after that, these cells start making a harmful poisonous protein. And that protein starts spreading in whole body. That is called actually cancer. And that is called tumor. Clear?
Oh, okay, sir.
Cancer is nothing. Just it's the definition of cancer is uncontrolled division of the cell is called cancer.
Okay, sir. Thank you.
And we will study, welcome. We will study in detail in class 12th. You have especially a topic, what is the benign cancer, what is the benign tumor, what is a malignant tumor. I will explain you in detail what is the carcinogens, what is the gene, which oncogene, which causes the cancer. We'll study in detail in 12th class, right? Fine. So last but not the least, that is the thrombocyte or blood platelets. So next one is thrombocytes or blood platelets. Blood platelets or thrombocytes. Again, we have already seen the structure of these blood platelets, right? So we have seen already the structure, their structure, and they are simple kind of the cell. Yes, diagram we have already discussed, and let me show you again the diagram of these. So this was the diagram of the blood platelets, right? These are the platelets, and they are also responsible for maintaining the immunity of our body. They are also called thrombocytes because they are responsible for the blood clotting in our body whenever we get injury. So they are responsible for blood clotting. So please note it down. Normally, the number of platelets, are 1,50,000 to 3,50,000 per mm cubic. Next point. Increase in number of platelet called thrombocytosis. Full stop. Decrease in number of platelet called thrombocytopenia. Full stop. They are rounded, oval, or disc-like bodies. They are rounded, oval, or disc-like bodies. Full stop. They are colorless. They are colorless and do not have nucleus. They are colorless and do not have nucleus. That is non-nucleated. That is non-nucleated. They are colorless and do not have nucleus. That is non-nucleated. Right? So this was all about the again some functions. Please note it down, some of the functions. So if we talk about the functions of platelets, so blood clotting is the main function of platelets. When we talk about the function of platelet, so the main function is blood clotting. So they are responsible for blood clotting and note it down. Their main function is blood clotting. Full stop. Their lifespan is a week. Their lifespan is a week. One last point. Formation of platelets takes place from megakaryocytes of bone marrow. Formation of platelet takes place in megakaryocyte cells. Formation of platelet takes place in megakaryocyte cells of platelet. Is it clear to all of you? Okay.
Yes, sir.
Now the next point is blood group. What is the blood group and what is Rh positive, what is Rh negative? So let me discuss with you what is the blood group. Blood group. So first, who discovered the blood group? Who discovered the blood group? Note it down, please. Basic but important. The blood group A, B, and O. These three blood groups. The blood group A, B, and O was discovered by the blood group A, B, and O was discovered by Ladsteiner. Ladsteiner. The blood group ABO was discovered by Ladsteiner in 1900. Full stop. AB was found out. Now the person who found the AB blood group. AB was found out by De Castello. AB was found out by De Castello and Stanny. So this was the basic thing. Now let me tell you what is the blood group, right? So there are four kind of the blood group generally which are present among us. So A, B, AB, and O. So the name of the blood group is based on antigen which is found on their surface. And here we are going to study antibody. So can anybody tell me, before going into the antigen and antibody, I want to basically know what are the antigen or what is the antibody? Any one of you? Can any one of you give me an idea what are the antigen and what is the antibody? Nobody have idea? Antigen? Look. What is the antigen? Antigen, the definition of antigen, if you note it down, you can note it down. Any external substance which is not the part of our body and which is not the part of our body, which is not in the composition of our body, and against when it enter in our body, when it enter in our body, and our WBC start forming antibodies against it, is called antigen, right? Or our immune system respond against it. For sake of example, the antigen is not fixed. Antigen may be biological, antigen may be non-biological component, right? So first, let me make you more clear. For sake of example, the definition of antigen is not fixed. If you have allergy with the smoke, means this is antigen for you. If you have allergy with the smoke, means it is antigen for you. But if I do not have allergy with the smoke, it is not antigen for me. So the same thing, particular thing may be antigen for you, but not for me. If you are allergic to the dust, means this is antigen to you. If you are allergic with any chemical, it means it is antigen for you. You must have read out the information which is written out of the hair color in the hair color box, that you may be allergic for this chemical. Before coloring, please try it at any part of your skin. Definitely you will see that. Some of the people, they are allergic to hair color and some of the cosmetic substances. That may be the allergen. That may be antigen for you. If you are allergic for that, that is antigen. Again, the antigen may be a biological object. Why? Like bacteria. Bacteria. It may be virus, right? It may be protozoa. It may be fungi, fungus. It may be pollen grain, right? They all are biological. So antigen may be biological or non-biological. Whenever, for sake of example, there is a bacteria which is antigen. When a bacteria enters in our body, what will happen? WBC will start searching them. When WBC will found that these are the antigen, what will happen? Some of the WBC will start secreting a particular type of the protein against it. That when the protein will come in the contact, it will kill the bacteria, right? So this protein, this is the protein which has been secreted by the WBC. What we call that protein? This protein is called actually antibody. So there are two means by which WBC can kill bacteria or any pathogen. So these are which antibody. So those WBC which secrete antibody, they kill the bacteria, but they do not eat bacteria. So they are called non-phagocytic. Non-phagocytic. Again, there are another kind of the WBC. They do not believe in secreting any kind of the antibody like substance. What exactly they will do? But they have also to kill this bacteria. So what exactly they will do? They will go to bacteria and they will just attack and they will eat bacteria and they will die themselves. So they are soldier of our body. They will eat bacteria. They will eat antigen. They will eat antigen and they will die. These kind of WBC are called phagocytic WBC. Phagocytic WBC. So there are two types of the WBC, phagocytic and non-phagocytic, right? So is it clear? Antigen and antibody clear or not? Please tell me.
Yes, sir.
Crystal clear?
Yes,
Please
sir.
note it down. Please note it down. Then I will tell you the blood group. Done? Okay. Look, if the blood group, so what I told you, I have told you that the name of the blood group is basis on the antigen. If any blood group is A, then this is blood group. So if blood group is A, then it will have A antigen and the antibody will be B. If the blood group is B, it means it will have B antigen and antibody will be A. If it is blood group is AB, then it will have A and B both antigen but no antibody, right? No antibody. That's why they do not have any antibody. That's why they are called universal. They are called universal recipient. So the person with the AB blood group, they can take blood group from any other blood group, right? Now it comes to the blood group O. The blood group O, it do not have any antigen. It do not have secrete any antigen. So, but they have antibody A and B both. So that's why they can donate blood to rest of all the blood groups, right? That's why they are called universal donor. Clear to all of you?
So like,
Yes,
what are these,
sir?
like A antigen and B antigen?
So they have both kind of antigen, AB antigen, right? Those A and B antigen. So, right, if you will give blood group AB blood group to A, they have B antigen, right? Means they will start, like they will the formation of antibody will start, right? So suppose that I am a person with A blood group. If you will give me B blood group, then my blood group A will start making antibody against it, right? If you will give me AB blood group, then the B is okay, A is okay, but AB have both of the component A and B. Then the B antibody will start working on me, right? So both are useless for me. But if you will give me O blood group, means there are no antigen, right? So my body will start not making antibody against the blood group O. Yes. Any more question? Somebody was asking. Someone was.
Sir, is O negative also a universal donor?
Yes, yes. O negative is the universal donor. I am going to tell you what is the negative and positive concept, right? Is it clear? A, B, A, B.
Yes, sir.
Okay. What will happen if suppose that a person have blood group A and you are going to supply him B blood group or AB blood group? What will happen?
So like the person will cough and throw blood. Like
person will die. Definitely
yeah.
he will die. He or she will die. Why? If you will
I
give.
saw that in the surgery. It was the patient
Sorry?
was throwing blood.
You
I've seen in surgery. Yeah, like the patient was throwing blood, like coughing.
Yes, yes, yes. They will start coughing blood and all that, like because the capillary will rupture, right? So what will happen? Actually, if a person have A blood group and you are going to give him or her B blood group, the A and B blood capsules will start clumping. They will get stuck. They will start clumping, and the process is called agglutinization. And after the clumping, the blood will become thick, and they will block arteries and veins, and the blood veins or blood vessels will rupture. This is called hemorrhage, right? And the person will definitely die, right? If you have given a sufficient quantity of the blood, person will definitely die, right? So that's why we cannot give them a wrong blood group, right? The next thing is that
So if the doctor knows that a person is having B or for example B blood group, then how can they give him A blood group?
Not possible, practically not possible. You know, means I would like to say he is not doctor. Right? Even the people who are pathologists, even they have the idea, this idea. Even those who are like even those who are a good nurse or compounder, even they have the idea. A very basic kind of concept, right? Even those people who do the course of nursing, even they have they get the idea of that what kind of the blood and which kind of the blood and what situation it should be supplied, right?
Okay, sir.
Okay. So next is, what is the Rh positive, what is the Rh negative, right? What is the positive and negative? Why we call that I have a positive blood group? Why I say that I have negative blood group? What is that? Look. So the person who discovered the Rh blood group, so why we call Rh? So this is called the person if have Rh factor. So what we call, look, this is called actually Rh factor. What is actually Rh factor is? First, I want to tell you what is Rh factor. Suppose that this is a RBC. Out of the surface of RBC, some of the people, some people have a particular kind of the protein on the surface of this RBC. This protein is called actually Rh protein, right? Or you can call it Rh factor. The name of the Rh factor come from, actually this has been discovered in rhesus monkey. There is a monkey where we do all experiment and we try all medicines made for the humans first. We do the experiment on the monkeys. So it has been discovered in rhesus monkey. That's why we call it Rh. Rh means rhesus monkey, right? So the protein discovered in rhesus monkey. The person who discovered it, his name was Ladistiner and Webner, discovered by Ladistiner and Wiener, W-I-O-N-E-R, Wiener, right? Ladistiner and Wiener discovered this Rh factor, right? So Rh factor was discovered by them. So if the Rh factor is present, then the person is called with a positive blood group, right? If Rh factor is absent, then we'll call that person with the negative blood group, right? So that is the mean of negative and positive blood group. Clear?
So can you explain?
Yes, sir. So, sir, the absence of
Yes. Absence of
Rh factor or Rh protein is very rare. That's why the negative blood groups are rare.
Yes, yes, most of the people have Rh factor on the RBC. So most of the people have Rh protein around their RBC. That's why this is called Rh positive blood group. Yes. O negative is rare. So suppose that I have, suppose that you and I, we both have A positive, A blood group, right? And my blood RBC capsules, they have a protein on the surface of my RBC, Rh protein. So I'll be Rh positive, or you can say you are A positive. I am A positive. And if you have the same blood group, but you do not have Rh protein, then you will be A negative. Clear?
So what is the use of this protein, sir?
Nothing. That's in evolution, it might have some function, but not now. Having the Rh positive and having the Rh negative, there is no merit or demerit of this protein. Just this form. Clear? Positive, negative, clear?
Yes, sir.
So, sir, people with negative blood groups cannot take
negative blood group. Sorry, people of negative blood group.
People of negative blood group cannot take positive blood.
Not take. You know what? Why? Because suppose that you have Rh negative, means we both have the same blood group. You have A blood group and I have A blood group. We both have the A blood. But only the difference is that you are A negative, I am A positive. So suppose that if you take my blood, what will happen when my blood will go in your body? So your body will start making antibody against the Rh protein, because now your body will treat Rh protein as a antigen. Clear? So the formation of antibody will start in your body, and your antibodies will kill all the RBCs of my body. Whatever you have taken from me, that will be killed. Is it clear? So, but the
Yes,
negative, the person with, but I can take blood from you, because if I will take blood from you, there is no Rh protein, means antibody formation will not take place in me. So why O negative is universal donor? Because O negative can give blood to O positive as well. Rest of the group and O positive as well. O positive can give blood, all of the blood groups, but it cannot be given to the O negative person. Is it clear?
Yes, sir. So, sir, O positive cannot give blood to
O negative.
What about is there A negative as well?
Yeah, A negative is also found. Yes.
So O positive cannot give blood to A negative and B negative as well.
No.
Okay.
So that's why O negative is considered as universal donor, right? Fine, 100% clear.
Yes, sir.
One more important thing which I want to tell you, please be focused. We will discuss again this concept in class 12th in human reproduction, right? So, for this year, I want to tell you a very important factor and very important concept, and you have to be focused over here. You will get these questions in medical examination. Even your school authority can ask this question. Look, there is a disease called HDN. HDN disease is hemolytic disease of newborn. What is that? Let me tell you. Suppose that there is a mother. Mother have negative blood group, right? Now what will happen? A mother have first baby. So first baby will have positive blood group, right? What will happen? Mother body will start making antibody. But the first baby will survive, right? Now again, the same mother of second baby, right? Second baby is also with positive blood group. Now the mother already have antibody against the positive blood group. These antibody will kill second baby. And this disease is called HDN, right? Hence, in previous older days, a mother cannot have two positive blood group baby, right? First baby survive, even it survive, but it have jaundice like situation, it will have pneumonia like situation, right? If it will not be completely or healthy baby, first baby. But the second baby will not be even survive. The second baby, the body of mother will start treating second baby or second fetus as antigen, and it will kill that, right? This is called hemolytic disease of newborn. Is it clear?
Yes, sir. So why will the first baby survive? Like if this
actually, because immediately formation of blood, transfusion of blood do not takes place between the mother and the baby. There is no connection of blood with placenta. Transfusion of blood do not take place between mother and baby with the help of placenta. Transfusion do not take place, right? Then the formation of antibodies very slow, very slow. Clear?
Yes, sir.
That's why the first baby will survive, right?
But so if please, the first baby can survive, then why?
Yes, first baby will survive and second baby will die. I will dictate. Please note it down. Okay. If the fetus is Rh positive and mother is Rh negative. If fetus is Rh positive and the mother is Rh negative, first baby will survive, first baby will survive, but in second baby, it will lead to, but in second baby, it will lead to hemolytic disease of newborn. Hemolytic disease of newborn, and it won't survive. But in second baby, it will lead to hemolytic disease of newborn, and it won't survive. So the blood concept is clear?
Yes, sir.
Now let's discuss with what is that? We did it. Okay. So now there is a topic of blood clotting. It's a very important topic. Blood clotting. Yes. Before going to the blood clotting, yes. Before going to the blood clotting, I want to tell you what is the lymph, right? Then I'll tell you what is the blood clotting and blood clotting process. What is lymph? Can anyone tell me what is lymph? Put the heading lymph. What is lymph? Come on, guys, tell me. Look. If you want, then I will tell you what is lymph. For sake of example, this is a blood vessel. It's a blood vessel. It may be blood capillary, blood artery, blood vein. It is blood vessel. And inside blood vessel, there are look, this is blood vessel, and inside blood vessels, there are some like blood is flowing in these vessels. But you know, these blood vessels are not completely impermeable. And these, look, one more thing I want to tell you that for sake of example, we have tissue. This is a tissue or cell. Suppose that this is a cell. You have a cell. So it's a cell. This is a cell or tissue. Now, blood capillary do not remains in direct contact with these blood cells or blood capillaries, right? Now, what happened? From these blood vessels are not completely impermeable, right? So some amount of blood plasma leak out over here, right? So leak out of over here, and it keep on accumulating in the space between the capillary, or it space between the capillary and tissue, and space between the cells, right? This leak out substance is called lymph. So lymph is a yellow color substance. Along with the lymph, what happened? There are certain cells that are WBC cells. We have discussed this. So these are the lymphocyte cells. Out of WBC, lymphocyte cell also comes, right? So this liquid is called lymph. Lymph is known as middleman. Why we call it middleman? Because the transfer of substance between the blood capillary and cell takes place with the help of this middleman. If this blood capillary have to give oxygen, then oxygen will come first to the lymph, and lymph will give it to these cells. And if the cell have to transfer the carbon dioxide will be released in the lymph, then lymph will give it to the blood capillary. So if the cell want to take the nutrition, it have to take through the lymph. If cell have to give the excretory product, then it have to give with the help of lymph. So that's why the lymph is called middleman. And there are some lymphocyte cells. They are a type of WBC. Why they are? Because if in these spaces there are any bacteria, there are any virus, there are any protestant, then these WBC or these lymphocyte cell will eat and kill them. Is it clear or not? Please tell me.
Yes, sir.
Fine.
Sir, this lymph is like the blood plasma?
It's a blood plasma, but some component of the blood plasma are missing in the lymph because it gets filtered out with the help of these capillaries. Clear?
Yes.
But it's a part of blood plasma. Please make this diagram, then I will explain some point for you. So lymph is made up of two things. Lymph cell, lymph is made up of lymph plasma, lymph cells, or capsules, and lymph plasma. And lymph capsules are mainly leukocytes, lymphocytes.
So, sir, lymphocytes come from WBC?
Yes, they come from the, yes, they are a WBC. They are a type of WBC. We have discussed even before.
So, sir, like lymph acts as a medium for movement of molecules.
Yes,
Yes,
it has medium. So if lymph will keep on accumulating over here, our body will swell. So there is a system which help us to again pour back these lymph. So there are some another capillaries or vessels. These are called lymph vessels, right? They are called lymph vessels. So there are another pipeline parallel to our blood vessels. They are called lymph vessels. So what these lymph vessels do, these lymph vessels pour back again the extra lymph to blood capillaries. Hence, the volume of blood always remains maintained. It do not get reduces, right? So these are called lymph vessels. Lymph vessels have some node-like structure in their capillaries or in their vessels. These nodes are called lymph node.
Sir, can you please explain the lymph vessels again?
Yes, there are another kind of the blood vessels, right? So there are another kind of the vessels will pour back. They collect the extra lymph from these spaces. So because lymph is keep on coming from these blood vessels. So if it will keep on accumulating, our body will swell, right? Because of accumulation of these liquid between our tissues, right? So what happened? These extra lymph is collected by these lymph vessels, and they again it drop or pour back in the blood vascular system. That's why the content or quantity of blood remains constant. Clear?
Yes, sir.
Else the blood quantity, we have 5.5 liter blood only. Now the if keep on if these lymph will keep on leaking out, then what will happen? Someday we will lose our all the blood, right?
Sir, so if these lymph veins are present, sir, then why it's accumulating here, sir?
Why
it's accumulating between the cells and the blood vessel?
it
like if
remains
the lymph
filled,
veins are
it remains
present.
filled up to certain amount, right?
Yeah. No, why is it accumulating if the lymph veins are present, sir?
Lymph, they just take out extra lymph, not all lymph. They just take extra lymph. because there is a certain space. So lymph will fill over there. A certain space is over there. When extra lymph will come out, then what will happen? Pressure will create in our outside. Then what will happen? Because of this pressure, this lymph vessels will collect all the extra lymph and they will pour it back. That's how it goes. And lymph is required over there because it works as a middleman.
So, sir, is there any condition where lymph is not leaking out?
No, there is no condition. But there are some conditions when lymph get accumulated. When you we get injury, what happened? I just suddenly fell down. What will happen? My hand will swell over here. Yes. So
Yes,
why
sir.
it is swelling? Because lymph is accumulating over there. You must have listened a name of a disease called elephantiasis, or the disease is called elephantiasis. We call it filaria. Yes.
Yes, sir.
What happened on that disease? Lymph start accumulating in legs of a person, and the leg become a thick like an elephant. That's why called elephantiasis, right?
Yes, sir. So anywhere where our body is swelling, so extra lymph is being leaked out.
Exactly. Yes, extra lymph getting accumulated over there. Clear? Note it down.
Yes, sir.
So please start noting down some point. Lymph is a mobile connective tissue. Lymph is a mobile connective tissue composed of lymph plasma. Lymph is a mobile connective tissue composed of lymph plasma and lymph cell. Full stop. Lymph plasma contain protein, calcium, phosphorus. Lymph plasma contain protein, calcium, phosphorus, high glucose and antibodies. High glucose and antibodies. Full stop. Lymph cells are amoeboid WBC. Lymph cells are amoeboid WBC. Mostly they are lymphocytes. Mostly they are lymphocytes.
Can you please repeat this one?
Lymph cells are amoeboid leukocytes. Mostly they are lymphocytes. There are certain organs which secrete lymph as well. Please note it down. What is the name of those organs? There are certain organs which secrete lymph. There are certain organs which secrete lymph. They are lymph node. I have told you what is the lymph node. They are lymph node, tonsils, thymus gland, and spleen. Thymus gland and spleen. One last point. Please remember the name of these lymph organs which secrete the lymph. It's an important question. Lymph work as middleman between body cells and blood. Lymph work as middleman between body cell and blood, comma. They destroy microorganism. They destroy microorganism. That's it for the lymph. Is it clear?
Yes, sir.
Okay.
So, sir, any kind of swelling, like swelling due to any exertion, like, so that is also due to lymph only?
Yes, that is due to the lymph one. That is due to the lymph collection. Swelling takes place in our body due to collection of the lymph one. So what happened? Doctor, what doctor says? If you have a swelling on that any part, what doctor says? What generally you do?
You put ice.
Doctor says go to home, have a water and salt, hot water and salt. Put your leg over there. Put your feet over there. Right? What they say? Just
yeah,
go.
sir. We put our hand in like hot water.
Yeah, right. Hot water, hot salty water, and they put the salt over there. And they says that have some warm cloth or have some cloth and warm it with the help of some warmer and put it in your leg or hand, whatever, wherever the part is swelling is over there. So what happened when you put your leg in that hot water, hot salty water? Salty water is concentrated water. Exosmosis takes place, so lymph releases the water. Water come out from the lymph, and swelling become less. When we put any hot object over there, that part, what happened? Because of the hot water, because of that hot temperature, water come out of the lymph, then swelling reduces. Simple science. Now the next topic is blood clotting, right? Blood clotting. So is it good or bad for our body? And blood clotting is also known as blood coagulation. It is also called blood clotting or blood coagulation. C-O-A-G-U-L-A-T-I. Coagulation, yes. C-A or C-O-A. Yes, blood coagulation. So look, what is actually blood clotting or blood coagulation? Whenever we get injury at any part, and what happened? Suppose that I fell down on ground and I got some injury. Blood start oozing out. After a few minutes, what will happen? The blood will become thick and it will get clot. Thick substance. It will become a thick substance. It will get clot on that injury, and it will block that opening, and it will the extra blood will stop coming out of my body. So it will help me out to lose extra blood. That is called blood clotting. Clear? That part is called blood clotting, right? So how the blood clotting or blood coagulation takes place? When I started the topic, I told you a diagram. Did I? Yes. I saw you this diagram. I explained you this diagram, right? So this was a blood clotting, right? In blood clotting, I saw you that if a blood is kept over there, free oxygen or molecular oxygen is coming. In 6 to 7 minutes, average, maximum 20 minutes, the blood clotting will start. Blood will change into color. It get divided into two parts, blood plasma and blood clot. This clot will make a thick covering for the injury, and blood clotting will take place. But on that time, I told you that I will explain in detail what is the blood clotting exactly and how the blood clotting takes place, right? So blood clotting clear? What is blood clotting?
Yes, sir.
Now let me explain you what the chemical reaction which takes place during the process of blood clotting. I will show you with the help of a flow diagram and you will understand each and everything. Focus over here. Let me explain for you. How does it occur? So first, what happened? We get injury So tissue get injured. So injured tissue. Like it may be our muscles are forward. Injured tissue. What they release? They release a certain kind of the substance that is called thromboplastin. They release a certain substance, and name of the substance is thromboplastin. So they release a substance, and that is called thromboplastin. So thromboplastin is released. Thromboplastin in presence of calcium and some protein. So in presence of calcium, thromboplastin in presence of calcium and protein. It will form an enzyme that is called prothrombinase. Prothrombinase enzyme formation will take place.
So what is thromboplastin?
Thromboplastin is a type of proteinaceous substance. Proteinaceous chemical. Prothrombinase enzyme. Right? So prothrombinase means pro means it is an active enzyme. So this was the reaction which takes place due to the tissue, right? Now what will happen? Another reaction is also taking place parallelly. If we'll get injury, the blood platelets will disintegrate because they will also die. Blood platelet disintegrate. And what they do, these blood platelet, they also release a substance. They releases platelet factor 3. They releases platelet factor 3. This platelet factor 3 is also called thromboplastin, or we also call it platelet thromboplastin. So this is the same thing which is secreted over there. So what happened? This platelet thromboplastin in presence of calcium and protein, it will make the prothrombinase enzyme. What prothrombinase enzyme do? So there is a hormone we have studied. The name of the hormone is heparin. Heparin was anti-clotting agent. Means heparin help not to clot the blood, right? What prothrombin do? Prothrombinase, it inactivate heparin. It inactivate heparin. Heparin, remember, anti-clotting agent, right? Heparin.
Yes.
And catalyzes a reaction. Catalyzes reaction. What reaction it catalyzes? What it do? What actually it do? Now this prothrombinase enzyme, it convert prothrombin. So prothrombin is inactive protein. Prothrombin. It convert prothrombin into thrombin protein. And it convert prothrombin into thrombin, and thrombin is type of protein. So prothrombinase will act as catalyst, and it will convert prothrombin into thrombin. Means it convert an inactive form into active form. Again, the thrombin act as an enzyme. Now this thrombin will act as enzyme. This thrombin protein. What it will do? It convert fibrinogen into fibrin. Fibrin is also a protein, fibrous protein. Now, so here in presence of thrombin. Thrombin act as catalyst. Now, what will happen? These thrombin and fibrin. These thrombin and fibrin both will accumulate above the injury, and these are the both two substances which make blood clot on injury, and the blood will stop oozing out. Is it clear or not? Flow diagram is clear?
Sir, can you please explain the last part?
Yes, I'll explain it again. Look, be focused over here. When we get injury, tissues releases a substance, and the name of the substance is thromboplastin. Thromboplastin is a type of chemical. Again, this thromboplastin in presence of calcium and protein changes into prothrombinase enzyme. Again, in another place, that blood platelets disintegrate at injury point. Blood platelet disintegrate. The releases platelet factor 3. Platelet factor 3 is same thing. This is platelet thromboplastin, right? Again, this platelet factor 3 or platelet thromboplastin in presence of calcium and protein changes to prothrombinase enzyme. Prothrombinase enzyme, what it do? It inactivate heparin, because heparin will not allow the blood to clot. So first it inactivate heparin, which is found in already present in our blood, and it catalyze a reaction. What it do? First it inactivate heparin, and the second thing what it do? It changes prothrombin. It changes prothrombin into thrombin protein. Now the thrombin protein is a clot protein, and that also work as an enzyme. This thrombin protein changes fibrinogen into fibrin. So thrombin and fibrin, these are two substances which make a clot on the our injury, and they prevent the extra blood to come out. Is it clear now?
Yes.
So please note it down.
Sir, where did this fibrinogen come from, sir?
It is already present in our blood. Fibrinogen is inactive, a fiber-like protein. Prothrombin is inactive, fiber-like. It's a clot-like protein. It is already present in our blood. These both proteins are present in our blood, in blood plasma. Right?
Yes, sir.
Please note it down. This is the flow diagram. If your school authority asks you to tell me what is the blood clotting process, draw first please flow chart and then explain it same in your notebook.
So what is platelet factor 3?
Platelet factor 3 is same thing, thromboplastin. I have written over here. That is the same thing. Platelet factor is platelet thromboplastin.
So what's written under platelet factor 3?
Below this is the platelet factor, the platelet thromboplastin. The same thing, this one.
Sir, can you scroll a bit down?
Yeah, sure. Done.
So prothrombin and fibrinogen, they both are responsible for blood clotting, right?
Thrombin and fibrin. Yes, you can say prothrombin and fibrinogen as well.
Okay, sir.
Because pro means inactive. Try to understand. If pro is written before any enzyme, means inactive. If fibrinogen, ogen, if suffix, this suffix is there, if this prefix is there, it means inactive. Look, if this kind of prefix is there, or if this kind of suffix is there, it means these are the inactive. Prothrombin is inactive enzyme. Thrombin is active, right? Fibrinogen is inactive and fibrin is active. Clear? That is the only difference between fibrinogen and fibrin and prothrombin and thrombin. So thrombin and fibrin, these make a layer-like gel-like structure or layer-like substance, which stop the oozing out extra blood from or any wounded part. Done?
Yes, sir.
Okay. Please put a note over here. There is a disease which is genetical disease. We will discuss this disease in detail in the our 12th part, where we'll study the molecule, the Mendelian genetics. In Mendelian genetics, I think it's chapter number 4. So there we will study this disease in detail. Right. Till the time, I want to tell you, hemophilia is a disease where blood clotting do not take place in the body of patient. This disease is also known as royal disease, or this is known as bleeder's disease. Also, the another name, bleeder's disease, and this is a genetical disease which comes from our parent. This is a genetical disease. Hemophilia is the disease in the patient, hemophilic patient, blood clotting do not take place in their body. Clear?
Yes.
Should we move ahead? Fine. Let me discuss you some more concept. So now come to the circulatory part. This was the body fluid, and now we are going in the part of circulation. This chapter name is body fluid and circulation. We discussed all of the aspect of regarding the body fluid. Now we are going to discuss with the blood circulation part. So how does the circulation of this blood or these fluid take place in our body? So first we are going to discuss the double circulatory system. I think this is the circulatory pathway. Okay, before going to that, I want to tell you what is the open and closed circulatory system. Did you know that? What is the open and closed circulatory system? You must have studied if you have covered, like if we have covered Animalia, in Kingdom Animalia. Do you have any idea what is the open and closed circulatory system? Look, there are two types of the circulatory system in animals. So what happened in open circulatory system? In open circulatory system, there is no artery or vein. Blood remains filled. Blood remains filled in spaces called lacuna, right? And organ bath in blood. Organ keep on floating in blood. They may or may not have heart. May or may not have heart. Now I want to show you what is the open circulatory system. For sake of example, it is found in insect where open circulatory system goes. Example, arthropoda. That is insect. So for sake of example, this is an insect or this is an arthropod. This is an arthropod. Right. In this arthropod, the body of arthropod, they have certain chamber-like structure in their body. So there will be some spaces, some chamber-like structure. They have a certain chamber-like structure. And these chamber, there are organs are present. Organ remains present. So example, it may be heart over there, some organ remain present over there. Right. And these organ remains bathing in the blood, and blood remains filled in these open spaces called lacuna, right? So these organ keep on bathing in blood. There is no arteries or vein like structure, and this is called open circulatory system. There are certain pores, they take the oxygen from here and they release the carbon dioxide from here, but blood do not goes out through these pores. So this is called open circulatory system. Is it clear?
Yes, sir. So, sir, all arthropoda have open circulatory system?
Exactly. They all arthropod, they have open circulatory system. Now, what about the closed circulatory system? In closed circulatory system, for sure, for example, all the mammals. Mammals have closed circulatory system. In closed circulatory system, there will be a heart, right? So there will be a heart-like structure, right? A heart-like structure will be present, right? A heart will have some closed vessels, like so. For example, this is some organ or tissue, organ or tissue. There will be certain kind of the blood vessels. Those are called arteries. So what artery will do? Artery will arise from the heart, and they are closed vessels. And these artery, they will carry oxygenated blood, right? So O2 or oxygenated blood. Now what will happen? This they will deliver oxygen, and they will pick the deoxygenated carbon dioxide, and they will deliver the carbon dioxide to the heart, right? So they will deliver. There are certain another vessels. They are called veins. Veins will deliver deoxygenated blood. Deoxygenated blood. So blood flow in. Blood flows in closed vessels. Vessels, right? So this is called closed circulatory system. Example, mammals, right? That is the difference between open and closed circulatory system. Clear to all?
Yes, sir.
Note it down. Open and closed circulatory system. Then we will study the double circulatory system. Done?
1 minute, sir.
Note it down. So please note down one more thing. So there is an important thing that all vertebrate means vertebrate are those which have vertebral column. So all vertebrate have muscular heart. So for sure, muscular heart will be present over there. Muscular heart. And that is divided into chamber. Muscular heart divided into chamber. So heart is divided in chamber. Out of them, Pisces means fishes. Pisces, that is fishes. They have two chamber. Again, we call about the amphibian. Amphibian have three chamber heart. Then it comes to the reptile, like the lizard and snake. Reptile have three chamber heart, but there is exception. Exception is that crocodile. Crocodile is a reptile, but it do not have three chamber heart. Crocodile have four chamber. Four chamber. And then it comes to the apes, in birds. They all are vertebrate. Then it comes to the apes. Apes, that is birds. Birds do have four chamber heart. And then it comes to the mammals. Mammals have four chamber heart. Right, please note it down. Crocodile is only exception in this chamber system. It's reptile but have four chamber.
Is there any other example of open circulatory system other than arthropod?
Yes, there are so many examples like this one. Annelids have heart and porifera, then annelida, they don't have circulatory system. Then nematoda. Yes, roundworm, they have open circulatory system. Roundworm, right? They have open circulatory system. Annelida, then echinodermata. Annelida have closed circulatory system. And echinodermata, they also do not have heart. Echinodermata is starfish. They also have open circulatory system. Note it down?
Yes.
Okay, fine. Now come to the next topic, that is double circulatory system. What is the double circulatory system? Put the heading, human circulatory system or double circulatory system. Then we'll, in next class, we'll study human circulatory, like human heart, how does the circulation take place? So next topic is double circulatory system. Look, this is double circulatory system. In double circulatory system, the blood takes two circle of heart. Look, what is that? There are four chambers. Out of this four chamber in our heart, like in human heart, so this is the atrium. So there are two atrium, left atrium and a right atrium. So these are two chamber, one and second. Then there are two ventricle, right ventricle, third, and left ventricle, fourth. Right? What happened? That deoxygenated blood is collected from, these are peripheral tissues and capillaries. Deoxygenated blood is collected from these veins, and with the help of veins, blood goes to the heart and it reaches to the right atrium. From right atrium, blood moves into, from right atrium, just a bit. Look, from right atrium, blood moves into deoxygenated blood. From right atrium, it moves into right ventricle. After right ventricle, this blood goes into this pipeline. This is called pulmonary artery. Look, the function of artery to carry the oxygenated blood. Artery carry oxygenated blood and veins carry deoxygenated blood, right? Deoxygenated blood. But here there are exceptions. Pulmonary artery is the only artery that carry deoxygenated blood, right? Now, the deoxygenated blood means carbon dioxide rich blood reaches to the lungs. It releases the carbon dioxide and takes the oxygen, right? Again, what happened? The pulmonary vein is the only vein which carry the oxygenated blood. Oxygen comes back. Oxygen reaches over here. The oxygen reaches over here. After reaching, what oxygen does? So oxygen reaches over here. Oxygen comes to the left atrium. From left atrium, it comes to the left ventricle, and then oxygen goes with the help of This pipeline, this is called aorta. Aorta is the largest artery. Largest artery. Now here the blood is taking two circle of the lungs. So here the first circle and this is the second circle. Look, this circle is called pulmonary circle because pulmonus means lungs. Pulmonary, right? And this circle is called systemic circle or systemic circulation. Systema means tissue because it takes to the tissue. So this is called double circulation, right? In double circulation, blood takes two cycle, blood complete two cycle. And double circulation. So why it is called double circulation? Because it takes two circle. The first circle is called pulmonary circulation. Pulmonary. And the second circle is called systemic circulation. Systemic circulation. And this kind of circulatory system is called double circulatory system. Please note it down. Please take the screenshot, and if you have any question, any doubt, please ask me. In next class, we'll do what is a human heart and how does the circulation takes place and disorder. And one more thing, electrocardiogram, ECG. So few topics are left, and we'll complete this chapter in next class. And in next class, even we will start the excretory system, and most probably we'll complete the excretory system by half. Half of the chapter we do it in excretory system as well. In case if you have any doubt, please ask.
So only pulmonary vein is the one which carries
only pulmonary vein is the only vein that carry oxygenated blood. Only pulmonary artery is the only artery that carry deoxygenated blood. These are the only two exceptions.
So sir, the ones we can see on our hands are arteries or veins?
Sorry?
Like the ones we can see on our hands, are
they
they
are the veins.
arteries or veins?
Actually, arteries are wound deep inside our body. You cannot see the artery. Veins are only peripheral and they are found in the outer surface. You can see only the vein. So those which are visible, like those color who have fair complexion and they are thin body. So you can see the green color of the vessels. These are the veins only.
Okay.
Any doubt, guys?
No, sir.
No? Shahid? All clear?
Yes, sir.
Okay. Shoaib, Nera, Shafullah, Nara? All clear? Fine.
Huh?
Yes, sir.
Yes, yes, please.
Sometimes we can see clotting inside our body, like due to some internal injury. So is that like how does it happen? Like, is it
Actually,
good or is it not good?
It's very bad. It's very bad. But you know, there are certain substances which dissolve this clotting. Right? It's bad. It's not good. Internal clotting is not good. That hamper the blood circulation. Right? And too much clotting may cause the even brain hemorrhage or any part of hemorrhage. Hemorrhage means rupturing of blood vessels. Right? Okay, guys. So see you in next class. And in next class, we'll start with the human heart and human circulation, then ECG, and then disorder. There are 4 topics are left in this chapter, and we'll start with the excretory system, right?
Yes, sir. So, sir, should we draw this diagram as well?
Yeah, you must draw it because you know your school examination. In a school examination, they will most probably they will ask you what is a double circulatory system. Then you have to draw this diagram. It's a very simple diagram. You have to draw this diagram, and you have to explain it, whatever I have explained. Right? Take the screenshot and draw it. Right?
Yes, sir.